Well, welcome back after lunch. So you guys get the dubious pleasure of listening to me 3 times today, and you're halfway there. So the good news is after this I'm done, so you don't have to listen to me talk anymore if you can just make it through. The other dubious pleasure is that I get to talk to you guys about instrumentation, which is so I'm an analytical chemist by training, and you know, instrumentation was my first passion as a scientist, I would say, and still haven't gone that far away from it. So if I get too geeked out or anything, then just say, Charles, tone it down, and we'll try to pull it back. So the real thing to think about here is what you're going to try to get out of this. Most of you guys are probably going to be using the core lab to run your samples for you. And so you may not need to know exactly how to run a mass spectrometer. And we're not going to teach you how to run a mass spectrometer here. You may not need to know, you might not even need to know exactly how a mass spectrometer works, as long as the data that it produces is useful. However, we still feel it's important to go over the instrumentation that we use to do metabolomics here, so that you have an understanding of how the data is generated, for one thing, just for your knowledge, but also so that you can understand the limitations, the strengths and the limitations of the techniques that we're using, and so that you can understand some of the terms that we use when talking with you about your data, and also importantly, so that you can read the literature and understand a metabolomics paper, a paper that uses metabolomics, understand the methods that they used, what type of things that tells you about the data type, and stuff like that. So that would really be my goal for you, is to be able to look at a method section from a paper involving metabolomics and be at least familiar with the terms, and know where to look and know how to think about what they did. Okay, so we'll define the objectives. We'll describe the workflow of the role of instrumentation in a typical metabolomics workflow. Why you might pick one instrument over another for a metabolomics analysis if you had the opportunity to make that choice, and then learn the key terms so that you could read a publication. All right, so outline. First, we'll talk about the tools that you would need for metabolomics. We'll briefly mention NMR versus mass spectrometry. Most of this talk is going to focus on mass spectrometry. Kathleen Stringer, who's going right after me, will tell you more about NMR and how that's useful in metabolomics and why it's really a very complementary technique. We'll briefly touch on why. So when we talk about mass spec in the context of metabolomics, usually we're talking about LC-MS or GC-MS. We call this a hyphenated technique, where you have 2 instruments coupled together. Why would we want to do that? And then Steffi will talk more about. Actually, she's very next. She will talk about chromatography and metabolomics and why that's a logical combination. And she'll talk a little bit more about this hyphenation. Sorry, forgot to take that out of my slide. We will not talk about chromatography methods overview. That'll be covered by Steffi in the next talk. Mass spec, we will definitely talk about methods overview. And then we'll look briefly at the instrumentation we have here at the MRC Squared. You'll get a chance to see some of that if you go for a lab tour tomorrow afternoon, and that's what we'll do. Okay, so instrumentation and omics science. This is one of those questions of, does the science drive the instrumentation to where it needs to go, or does the instrumentation, the availability of a technique, drive the science? And an analytical chemist would make an argument for the latter half. Maybe a molecular biologist would say the other way around. But really, both things are inextricably linked. You can't study something if you can't measure it, if you can't detect it. Sometimes you don't know what you need to be studying if you don't understand the biology behind it. So really, they're inextricably linked. And advances in instrumentation, in my view, have been really linked with the progress of science over the years and decades, especially in recent terms in the omics sciences. So the old way that we would look at things. So let's look at DNA, proteins, metabolites, this sort of central dogma axis of biology, if you can still consider it that. DNA, the old way that you used to have to look at DNA was Sanger sequencing, and very useful technique. You'd read out these long meters, long gels, and look for your code of DNA. The order on the gel would determine your sequence, and then you'd be able to sequence DNA. But it was very laborious, very time-consuming, and you could only read out so much at a time. Advances in instrumentation happened, and we went through several stages, and now we're at next-gen sequencing, which can generate a sequence in a matter of days or less. for an entire organism if you have the money and the resources to throw at it. And so this has really enabled a revolution in the way we consider genetics. Rather than looking at a single gene at a time, we look at the whole genome, and the instrumentation makes that possible. With proteins, something similar happened. We used to run 2D page gels, and these are still useful in some cases, and you can get a very exquisite separation of a complex mixture of proteins across a gel. But then you've got a gel, and what do you do with that? Well, you've got to cut a spot out and then carefully figure out what it is. Maybe if you didn't even have an instrument at all, a mass spec at all, you'd just be limited at approximate molecular weight and looking at how it performs, and then doing some other sort of assays to figure out what you had, and very challenging to do high throughput quantitation. Instrumentation came along, LCMS-based proteomics, shotgun proteomics, and we're able to do protein, much more comprehensive protein analysis. And initially that started in just detecting proteins, and then it moved to quantitation of proteins and protein turnover, and all sorts of other things have been enabled by proteomics. Something similar in metabolomics. Used to be the way you would measure metabolites would be by doing biochemical assays one at once. You'd order a kit, or you would buy your reagents, and you would get an enzyme that would measure, that would somehow lead to some downstream change in colors, and you would put that on a plate reader or a spectrophotometer, and you would measure your color, and that would be proportional to the concentration of one metabolite. And then if you wanted to measure another one, you'd buy another kit or get another enzyme and do the whole same thing. And that still can be useful. There's nothing wrong with that method, but it's very low throughput. And so Mass Spec has enabled this revolution, the ability to measure many metabolites at once. And of course, you already know that. That's why you're here. So let's say, what do I need to know about instruments? So here's back to my workflow, biological sample prep, instrumental analysis. And what we'll do today is we'll open up the black box and see what's inside for a little bit. And then once we're done, we can close it back up and put it away if you don't want to touch it again, or at least maybe you'll know something about what's going on. Okay, what tools are needed? Metabolomics, as I said before, is a study of all small molecule components of a cell, tissue, or organism. How are you going to address that, right? You've got many different compounds, a range of different concentrations. We might need to quantitate something that's very abundant, glucose, lactate, in a sample at the same time as we're quantitating something much lower, like a thyroid hormone or a steroid. And we're interested in all of it. We want to be able to look at it all. So we need an analytical method that's sensitive to be able to handle those low-level things. We need it to be quantitative. If you can't quantitate it, then why are you even going to bother with it? We need to be able to tell changes. We need it to be universal. We need it to respond to all sorts of different molecules, all classes. We don't want it to focus in on a particular property of a unique property of one particular metabolite, because then we won't be able to detect everything else. And it needs to be able to be capable of detecting many compounds simultaneously. We don't want something that's going to be analogous to a plate reader assay where we have to do a different manipulation for each sample. We want to be able to look at many things at once. And so the instrumental techniques which enable that, by and large, for metabolomics purposes, are LCMS, liquid chromatography mass spectrometry, GCMS, gas chromatography mass spec, and then NMR. And you could put a small asterisk in there and add CEMS, capillary electrophoresis. Some people like to do that for metabolomics. You could put in mass spec alone. Sometimes there are certain assays which can be done with a mass spec without a chromatography. You could put in, maybe under some circumstances, chromatography alone with an alternate detector. But by and large, most metabolomics is done on one of those three platforms. Okay, so we'll start just with a brief mention of NMR to sort of segue into Kathleen Stringer's talk later in the symposium. So NMR is a near universal detector. That's a great thing about NMR. NMR stands for nuclear magnetic resonance. It's analogous. You guys you may remember it if you ever had organic chemistry. You put it in the big, metal instruments. It's like a cryogenic flask with a probe, and you lower your tube in, and you get a spectrum out, and that's good for identifying your molecule. Well, it's the same technique. It's just applied in a much more comprehensive way, where instead of looking for the identity of one molecule, we're looking for many signals at once in the data. And I'm not an NMR expert. Kathleen can tell you a lot more about how that interpretation is done and how the software is done. But it's good because it responds to anything with a carbon atom or a proton in it, or any number of other different nuclei which are addressable by NMR. So it's very good for that. It's quantitative, very easily quantitative. It stays calibrated. Its response day one is very similar to its response 3 months or a year from now, as long as your instrument is properly maintained, without excessive effort. Mass spec we do good with quantitation as well, but there's a lot more work that has to go into it. Reproducible, provides structural information. As I said, NMR is routinely used for understanding the structure of a molecule. It requires relatively minimal sample preparation. Sometimes it's good enough to just dilute and load your sample into the instrument, or load it in as is, or do some limited protein precipitation, etc. NMR limitations, on the other hand, it's generally thought of as somewhat less sensitive than mass spec when you get down to the very low-level compounds. There's tricks you can play to improve that. Resolution is another thing. All of your signal pretty much comes out in a single spectrum. You can do 2D NMR to help improve the separation space, but you're limited in your ability to resolve things, and so there's some limit in terms of the number of total features that you can detect in NMR. NMR and mass spec are really complementary. You can get useful information by combining analysis done on both NMR and mass spec, as Kathleen can attest to and vouch for, and we'll tell you more about in her talk. And we'll have more on NMR later. So now let's talk more about mass spectrometry. Okay, so NMR versus mass spec. Okay, one more slide, sorry. So high sensitivity, this covers much of what I already said. Mass spec is a little bit better for sensitivity. Quantitative reproducibility, NMR is great. Mass spec can be good or bad depending on how you manage it. Mass spec does a little bit better with many analytes simultaneously, although NMR can handle quite a few. Generally high, close to 100 maybe, over 1,000. Structural information, NMR is great. Mass spec can be good depending. NMR is pretty good avoiding matrix effects. We'll talk more about that. Not so good mass spec unless you carefully manage. Can be coupled to chromatography. NMR can be done, usually doesn't work so well. Mass spec works very well. So we'll talk more about mass spec. Okay, very briefly on this hyphenated technique thing. Why not just do mass spec? Why put LC on the front? Well, LC, as Steffi will tell you, is good at separating complex mixtures. More about that soon. But it can be difficult to identify components from a chromatogram alone. Mass spec can determine the mass of molecules and is useful for identifying them, but signals from multiple components are difficult to differentiate where they're coming from. So when you put them together, you can get the best of both worlds. You can separate and then detect. This is what the typical layout of an LCMS or GCMS system looks like in a block diagram, sort of simplified format. You put chromatography on the front end, so you separate. Then you ionize your molecules, and we'll talk more about that and why that's necessary for mass spec. Then we analyze those ions by mass. This is the heart of the mass spectrometer, and then we collect our data. And so I'll show you. So Steffi will talk more about the chromatography side of things, and I'll be covering this in detail in this talk. So this is on an LCMS system. All of those components are here. We've got a LC system, and then we have the ion source where your ions are made, and then the internal parts of the mass spec is where the mass separation happens, and the detector is located inside as well. And then for a GC, the components are much the same. You've got your chromatograph going on essentially in a capillary tube inside an oven. Your ionization happens inside an ion source of the mass spectrometer. Your mass analyzer is in there too, and then you've got a data collector and connected to a computer. Okay, so that's it for chromatography until Steffi's talk. But let's talk about mass spec. What is mass spectrometry? Well, the process of mass spectrometry involves taking neutral metabolites or molecules or whatever it is you're looking at and turning them into ions. So why do we need to make an ion? Well, mass spec is a technique that works entirely on ionized molecules, and the reason is because it uses the useful property of ions that charged particles can be moved and manipulated in a magnetic or an electric field. If you have a neutral metabolite or a neutral molecule floating around, it's not going to be influenced by any sort of external field you apply to it. But when you get it charged, if you put a negative charge on a plate, you can get that ion to accelerate towards it if it has a positive charge. So the first thing we need to do to our metabolites is ionize them. Then our ionized metabolites need to be separated by mass, so we can measure their mass and resolve them one from another. Otherwise, we just have an ion detector. So once we've separated our ions by mass, we use a detector to read out, and hopefully that detector responds proportionally to the amount of each ion. So you can get a signal that's proportional to concentration. So Mass Spec detects any compound that can form an ion. If it can't be ionized, it's not going to work by Mass Spec. And there are relatively few metabolites that fit that criteria entirely. There are usually tricks that we can play for something that's resistant to ionization to get it to ionize, and we can talk about that if there's interest. Mass Spec is sensitive and selective. It's got high resolution, and Mass Spec can help identify metabolites and can provide some structural information when the criteria are right. Just knowing the molecular mass of a metabolite is a very useful piece of information because that really narrows down what it is that you're looking at. It's useful for quantitation. As I said, we wouldn't start if it wasn't, but the sources of interference must be carefully managed. You have to really know what you're looking at, and we'll talk more about that in a bit. Okay, so just by way of defining terms, this is probably familiar to most of you. The output of mass spectrometry is a mass spectrum, and that is a plot of intensity. So this is your abundance information versus mass to charge ratio. So what do I mean mass to charge ratio? So normally we think of a metabolite as having a molecular weight, which is its mass. Mass spec, as I said, is an ion detector. So you put a single charge or multiple charges. You could have a doubly charged, triply charged ion. If you have an ion that's singly charged, say it has a 1+ charge state, its mass to charge is going to be more or less the same as its mass to charge ratio because you have the mass and then you divide by a charge of 1. When you have a doubly charged ion, say something with a molecular weight of 1,000, if it formed a 2+ ion, you would expect to see that at a mass to charge ratio of 500. 1,000 divided by 2 would be 500. Actually, it's a little trickier than that because you put on, in the process of making an ion in electrospray, as I'll tell you in a second, you may add on a proton, which would add 1 extra Dalton of mass, or you might add on a sodium, which would add 22 Daltons of extra mass. So you have to consider how that ion is made in looking at the mass to charge ratio that you get. Okay, so let's talk about how you would make an ion, and this is relevant because it affects the signal that you get out of a mass spectrum. It affects what sort of metabolites we can see, and it affects those matrix effects that I'm telling you about. And so matrix effects, I should probably define that term for you, is anything that interferes with quantitation of your molecule of interest that is derived from something else that's present in your sample. And so ideally we would like an analysis with zero matrix effects where your signal is directly proportional to your concentration and there's no caveats on that. You could just directly use it for calibration. But in the real world, the other things present in your sample or in the environment can affect that relationship between signal and concentration, and we have to manage them. So electrospray ionization, as I was saying, is a way we make an ion out of your metabolite. And so what happens is typically, so this is only one of many, many, many different ionization techniques, and we're not going to talk about all of them. Electrospray is really, at present, it's really revolutionized the field. It's only been widely used for under 30 years. Since around 1990, it was sort of coming into its own as a technique and became widely adopted. But electrospray ionization is a technique that's very commonly coupled to liquid chromatography, and Steffi can tell you more about that side of things. But it works very well because it can make ions out of liquid, out of the liquid that's coming off of your chromatographic column. Almost everything can be separated by LC. There's a few things that can't, but it's a very good way to directly couple your separation method to mass spectrometry. And so we've got all these molecules in solution coming out of your LC column. We need to convert them into a gas phase ion. It wouldn't do you much good to have an ion that was solvated by a droplet of liquid because that droplet of liquid is huge compared to the size of the ion that you're trying to manipulate and measure. And so what we do in order to convert that liquid to an ion is we apply a high voltage to a probe that's at the outlet of your LC or whatever and the inlet of your mass spectrometer. And that establishes sort of a potential gradient on the surface of the droplets that are emitted. And through various processes, which even I am a little bit nebulous on, you form droplets that have a concentration of charge. The surface of the droplet has one charge. The interior of a droplet may have other charges. In addition to this, so you've got this charged droplet that forms because of this high electrical potential. You also add drying gas. So this is heated dry nitrogen usually, or potentially air that effectively helps the solvent molecules in this droplet to evaporate. And as that solvent evaporates, you're left with a smaller droplet, and you build up, the charge doesn't go anywhere. It builds up, and you get a more intensely charged droplet. And eventually you get to the point where there's so much charge in the droplet that it reaches sort of an electrostatic limit, and that droplet will then break apart into many other smaller droplets. And this is fission. This process is the Rayleigh limit, and then fission of droplets. And so eventually this continues to happen. You get smaller and smaller droplets, smaller and smaller numbers of ions per droplet, and eventually all of the solvent is gone, and you're left with exposed ions. And so the exact mechanism by which this takes place is still somewhat debated in the literature. I guess we're getting closer to a complete understanding of it, but not totally there yet. But suffice it to say, we produce gas phase ions. And the cool thing is we can produce gas phase ions for things that would normally never go into the gas phase. We could take something as big as a protein, which if you heated it up, it would burn up before you got it into the gas phase. But by doing this ionization method and by putting a charge on it, you can effectively have an unsolvated protein molecule entering that can be pulled into your mass spec. Okay, and so then you've got a charged particle. You can manipulate an electric field. You have some sort of a metal plate or cone or something that's your inlet to the mass spectrometer, and you draw those particles in. You also have this is all done at atmospheric pressure. Inside your mass spec is under vacuum, and the reason for that is if you take an ion and you just shoot it through the air, eventually it's going to collide with some other gas molecule and transfer its charge and lose its ionization. But if you pull it into a vacuum, there's nothing else there for it to collide with, so you can go on and manipulate it and work with it. Okay, so ESI. Let's talk a bit pros and cons. As an ionization technique, it's what's called a soft ionization technique. Means it doesn't decompose your analytes, even if it's a complicated molecule, something like a protein or a big metabolite. It usually doesn't break down in the process. It doesn't fragment, so you can get the intact molecule into your mass spec. It works for a wide range of analytes, from proteins to small molecules, from polar metabolites, works very well. Semi-nonpolar metabolites also work very well. If you have something that's truly nonpolar, you may have challenges, so we may have to use an alternate ionization method for that. Very efficient ion production. Efficient is a relative term, but you get a lot of ions for a given number, a given input. So it can be sensitive in that you can generate a lot of ions. Downsides, it works best when operated at relatively low flow rates. That's not a big deal for metabolomics. It's nice to be able to work with small samples. The analyte must be able to form a solution phase ion. If it can't form an ion at all in solution, then the odds that it's going to be ionized in the gas phase are essentially zero. It needs to be able to hold a charge at least for a short period of time in solution. Matrix effects, as I already mentioned, are the big downside of electrospray ionization. And the specific matrix effect that we have in ESI is called ion suppression. And so you may run into this if you're looking at literature about quantitation compensating for ion suppression. If you're developing a new method, you need to test for ion suppression and validate your method to show that your quantitation is valid in spite of this sort of thing. So ion suppression, what exactly is it? It's something that causes reduction in response of an analyte of interest, the thing that you're trying to measure, your metabolite, in the presence of an interfering substance. So something else in your sample. If you have a lot of salt in your sample because you've submitted cell culture media to the metabolomics core and you're trying to quantitate some low-level polar metabolite, we may have a problem because the concentration of salt is so high that it may be effectively the main thing that the mass spec sees, and you can't get down to that very low concentration that you need to be able to see your metabolite of interest. So you might need to do something to your sample in order to better prepare it for mass spectrometry. Some things that can cause ion suppression, as I mentioned, non-volatile buffers or salts, PBS rinses. That's super routine and very gentle in the context of biology, but it may not be the best thing to do if you're preparing to submit a sample where you're going to do trace level analysis for metabolomics. And so we can tell you some things about volatile buffers to use if you need to do a buffer rinse immediately prior to metabolomics analysis that can reduce that effect. Detergents are generally the enemy of mass spec. You might use Triton all the time in one of your cell extraction protocols for your biochemical assays, but that Mass Spec loves detergent. It loves to ionize detergent, and it loves it so much that it will prevent ionization of your other molecules of interest. So those are generally not ideal to have included. Again, there's ways around this, but just something you should be aware of. Other analytes present at higher concentration. This is one that's trickier because you can't just remove it from your sample prep procedure. You have to be able to manage it. So if you're trying to measure a low-level carbohydrate with the same mass as glucose in a method that puts all of your carbohydrates right around the same place in your analysis, then you might have a problem. And so we need to deal with that on the front end and on the separation context. So all of these things can cause ion suppression. It can vary from sample to sample. That's a challenge. If your matrix are different from sample to sample, you might have differential ion suppression. It could vary from run to run, day to day. Thus, it can cause problems with quantitation. So, oh no, why are we doing mass spec? Oh, well, one more example. Here's how bad it can get. This is an extreme case. It's usually not this bad. So this was a study where they were infusing a constant level of in this case, a drug into the mass spec, and then they injected a negative control sample, just the biological matrix, and looked at the signal from this drug, which should have been present at the constant level, and they saw, oh my gosh, it goes down 90%. So if you were trying to measure it down here and your calibration curve was off by 90%, you would be drastically underestimating. So why are we using mass spec for quantitation, and why do you guys claim that you can do quantitation? Well, there's ways around ion suppression that actually work quite well, and that make it so that in spite of this problem, which we need to be aware of, mass spec is actually very effective for quantitation. And so the first thing is to avoid adding components that can cause it. So when you're thinking about your buffer rinse, your last sample prep steps, you'll consult with the core, you'll look at our sampling guidelines, and you'll see things that are designed to help minimize this problem. If you can't get rid of it, we may still be able to work with it. So don't give up hope. But it's just something to be aware of. You can use an effective LC method, as Steffi will tell you about, to separate the molecules of interest from something that would interfere, and sometimes that's all you need to do, and it doesn't matter what else is in your sample. You can get a nice clean separation on the front end, and your matrix effects effectively go away. Another thing, a trick that we like to use and a lot, is to add a stable isotope labeled internal standard. And so this is different from what was mentioned about flux studies where you're using this to trace metabolism. We can just put something in that looks exactly like your metabolite of interest, but it's, say, carbon-13 labeled, and we can measure the extent of ion suppression and compensate for it. And then no matter how much occurs, if you put that same internal standard in all of your samples, you know the magnitude of your suppression effect, and your quantitation goes back to being great again. We deal with this on a daily basis, and it's something we're very comfortable with. And the good news is when properly managed, mass spec can and is extremely useful for quantitation in spite of these potential pitfalls. Okay, so now on to the fun part. Make sure my timing is good. So mass analyzer. So once you've made an ion, great, we've got an ion. How are we going to separate it by mass? As I mentioned before, ions can only be manipulated under vacuum. Hence, all of this stuff that we're going to talk about is done on an instrument under vacuum. And so when you walk into our lab, you'll hear the vacuum pumps humming. That just comes with the territory. Many different types of mass analyzers exist. You may see these in the papers that you read about mass spec. Magnetic sector was sort of where it all began. We've got quadrupole time of flight, and I'll give you a tour of the most relevant types of these detectors. Again, most of you won't probably need to pick one of these or another. The core lab will develop an assay or run an assay that we know works for your samples, but it's useful to know at least the terminology. So we'll start with the quadrupole. A quadrupole mass analyzer is sort of you can think of it as a selective mass filter. Not surprising, quadrupole has 4 poles or rods that are metal rods inside the mass spec, and you produce ions by ESI or some other method, and you stream those into the space in between these 4 rods, and you apply a direct current and a radio frequency voltage to these rods, and it turns out that, again, charged particles move in an electric field, and these ions start to oscillate. They take on a trajectory that depends on the voltages that you've applied, and all of this is done by a computer. Not even the analytical chemist really needs to know exactly how those voltages are worked. That's done by the engineers that designed the instrument. But effectively what happens is that at a given setting, this quadrupole can be set in such a way that only a single mass to charge adopts a stable trajectory and makes it all the way through these rods, these 4 poles. And other masses that are too high or too low, too big or too small, take an unstable trajectory and are ejected out of the quadrupole. And so then, and you can change these voltages and allow different masses to pass at different times, and you can read that out knowing what you get, and you can generate a mass spectrum. So the things that are of relevance, this is a low mass accuracy. We can resolve something that's about 1 Dalton, 1 mass unit apart from something else. So that's not so great. There's many things that have the same nominal mass but are different metabolites. But it can actually be very good for quantitation, and we'll talk about that in a little bit. Another type of mass analyzer is a time of flight. So this is kind of the most intuitive type of mass spec that you can think of. You take a bunch of ions, you put them in, you accelerate them all to the same kinetic energy, and then you just let them drift in a tube that's under vacuum. And so if they've all got the same kinetic energy, things that are smaller tend to move faster. KE equals 1/2 mv squared, if you remember your physics. So they go quicker. The bigger stuff moves slower, and it hits the detector later. And so what you get is you get an extremely accurate stopwatch here at the end, and you say, okay, I've got an ion arriving at 10 microseconds and another one arriving at 12 microseconds. You don't actually do that. The instrument does all that for you. And then that gives you that can then be calibrated and give you the mass that you're looking at. So smaller ions travel faster, as I already said. So you wouldn't think it, but it actually turns out that this is pretty accurate. So you can measure something down to, say, 2 or 3 decimal places and have pretty good mass accuracy. And so that helps you distinguish something that might have the same nominal mass but have a different molecular formula and give you some information about whether it's the same metabolite or whether you have 2 different metabolites or which one you're actually looking at. It also reduces the noise by helping you separate signal from background. It's a little bit more expensive than a quadrupole, and it requires careful attention to calibration. Okay, another newer one that you may hear about is an Orbitrap mass analyzer. This was a newer one, first introduced in 2005 by Thermo Fisher. Again, a lot of this stuff, if you ever get an occasion to buy a mass spec, you'll have to think about all of these different things. For us, we have certain instruments, and we choose the one to use that's most relevant. But the way this works, instead of moving ions through a field or a quadrupole, it traps ions in this interesting sort of toroidal-shaped trap, and these ions rotate around it based on the manipulation, again, of electric fields. And based on the pattern in which they rotate, different ions adopt different trajectories. It induces a frequency, and you can measure that frequency of the electrical signal that's generated by the ions in the rod, and you can do a Fourier transform if you like math, and then you can convert it to a mass spectrum. The output of the end result of this is you get a very high resolution mass spectrometer, and this is similar to an FT-ICR, which is sort of the original Fourier transform type of mass spec. But the output is you get even higher resolution. So 10,000 is pretty good mass resolution. This is a moderate resolution for a time of flight instrument. You can do 10 or 20 times better than that on an Orbitrap. So you can sometimes even distinguish things that were previously inseparable. The number of times when this actually happens in the context of metabolomics is relatively small, however. So generally, with the ability and the mass accuracy, in other words, the ability to measure the number of decimal places, is not all that much different here. It's just that you can do a little bit more fine resolution. The downsides of this is it's more expensive. Surprise, surprise. You buy a better measurement. There's also got more working parts, may have tendencies for more things to potentially fail. But that's not your problem. That's our problem. And we've gotten good at dealing with those things. Okay, so now to briefly touch on Tandem Mass Spec. So right now up to this stage, we've talked about a single stage of mass spectrometry. We make ions, and then we separate them by mass, and then we detect them. That's great. We can measure the masses of our metabolites. But what if we wanted more structural information? Or what if we had 2 metabolites that had the same nominal mass? Let's say citric acid and isocitric acid. If those have exactly the same molecular formula, if you put them in a mass spec, you're not going to be able to tell the difference if you just have a single stage of mass spectrometry. Well, it turns out we can do more. Some instruments have the ability to, once you're inside the instrument, take ions that are already generated and fragment them. And so one simple way to do that is to ram these ions into other molecules or species inside the mass spec. And so we might collide it with some sort of a gas like argon or nitrogen. And when you get these ions zinging in and ramming into other molecules, that imparts some energy into those ions, and it causes them sometimes to break. And so this basic technique is called collision-induced dissociation, or CID. Other methods of doing this exist too, and you get multiple fragment ions out. And so this is one metabolite here, and you can see that in this case we have a single mass, and it can break up in many different ways, and all of these different ions inside the mass spec can be detected if you have an instrument that's capable of doing it. And so not only do you get the ability to distinguish two things if they fragment in a different pattern, but you also can get some structural information by looking at the mass of these fragments. You know that, okay, I have an entity that gives me something with a mass of 174, 94, 74, and if you're an experienced mass spectrometrist, or if you have access to a database, you can piece this information together and get structural information. MSMS can give you good sensitive reproducible quantitation under some circumstances, but it does limit in some cases the number of metabolites that you can look at simultaneously, and we'll talk about that a little bit. Okay, so how are we going to do MSMS? Well, we need an instrument that's capable, and the most basic, most common one that's used to do MSMS is called a triple quadrupole, or a QQQ. So you may hear people talking about that. It's widely used. It's robust technology, and so basically you take 3 quadrupoles in a row. The first one is designed to do the mass filtering, as we already talked about. The second one, they call it a quadrupole, but these days it's actually usually a hexapole, is where your collisions happen, so where your ions collide with others, and this sort of just provides a guide for the ions and a space for that to happen. And the third quadrupole is another mass filter. So it's very good. I'll tell you, again, these figures of merit, these things are similar. It's the same as a quadrupole. It's got unit mass analysis. I'm sorry, it's not high MAC accuracy. It has unit mass resolution, and it requires some pre-analysis method development. So how do you use a triple quad? Well, you can do many different modes of operation. I put this in here because people sometimes ask when they're reading a paper, oh, that was done in SIM mode or MRM mode. What does that mean? Well, there's various different modes that you can use the instrument. You can run it in full scan mode, basically acting as if it was a single quadrupole where you detect all of your ions one at a time and detect all of your possible masses. That works, but it's not the most effective way to use a quadrupole. If you wanted to do that, you'd be better served by a time of flight analyzer. You can do selected ion monitoring, where you spend all of your time looking for one or just a few ions, and that improves the sensitivity of the instrument, because instead of spending your time scanning through all of these different masses, your detector can spend all its time looking for one or a few, just the metabolites that you find of interest or that you want to measure. And so that improves your sensitivity, which is great, but you're still not doing MSMS. So there's various different ways you can do MSMS. One way is to take a single ion in and fragment it and look at the fragments that you get out. So that would be to generate that spectrum that I showed you before, where you're trying to get structural information out of an ion, or see how a single ion. So your first ion, just to introduce another term, I know I'm inundating you with the term. The first ion is often called a parent ion. The second ion is called the ions that are produced after fragmentation is called a product ion. So that's a product ion scan. Sort of the reverse is a parent ion scan, where you look at multiple parent ions, you do a collision, and then you look for everything that gives a particular product ion that's of interest. And so if you had a class of metabolites that all have the same head group on them, then they might fragment the same way, and you might look at that using this sort of thing. The most, I'm sorry, this should be called multiple reaction monitoring, or select SRM or MRM. I apologize for that. That should read SRM and also MRM. Both terms are used somewhat interchangeably. So this is kind of a common mode of operation of a triple quad, and you'll see that in other contexts. You put a bunch of ions in, you select one of them, you do your collision, you get a bunch of fragment ions out, and then you select one or a few of them of interest. And that lets you really be more specific. So not only do we have the mass of the first one to define the ion, but we also have the mass of a particular fragment, which does two things. It improves your sensitivity by removing chemical noise, and gives you something that's selective to that metabolite of interest. So if you see a paper that talks about an MRM method, this is what they're doing. They're running a triple quad in such a way that they're targeting particular metabolites, and they're using this multiple stages of mass spectrometry to get that information. So all these different modes of triple quad, which I threw at you, make the instrument very versatile, but it does require more method development. and it's not as widely used for untargeted profiling. Very limited use. I've seen a couple papers where people have attempted it, but in general, that's better done on an instrument that's more widely used for profiling, like time of flight or something like an Orbitrap. So other types of tandem MS instruments, very briefly. A quadrupole time of flight is one of them, where we put a quadrupole that we've discussed already on the front end. We have a collision cell sort of same way, but then on the back end, we put a time of flight analyzer. And so the second stage of mass spec, now we're able to get an accurate mass, in other words, many decimal places for those fragments. And that's very useful for metabolite identification. It's a little bit less useful for targeted quantitation, and the reason for that is the speed at which you can do MS-MS is a little bit slower on a time of flight. You may not be able to monitor all the different metabolites that you're interested in when you use it in that manner. So that's why a TOF, when you use a TOF for quantitation, you usually use it in MS1 mode. When you're doing MS-MS in the context of metabolomics, it's normally being done for identification or maybe a very limited range of quantitation. There are some exceptions to that, some unique ways of using a QTOF, but by and large, that's still true. And Orbitrap is kind of much the same. We can have something called an LTQ Orbitrap, which is just another type of mass analyzer on the front end, which lets you select an ion and then an Orbitrap to get an accurate mass readout on the back end. Advantage of that, very high mass accuracy. Turns out this ion trap can do another trick, which instead of having 2 stages of mass spec, you can do multiple stages of MS. So you can fragment and then fragment a fragment and fragment a fragment a fragment. And under certain limited circumstances, that can help you even further with identification. Of course, this is among the most expensive type of mass spectrometers. Your throughput limited just by the fact that no one can afford enough of these to do many analyses a lot of the times, and more working parts more likely to break down. So if you ever have to do this, what's the best instrument for mass spec or for metabolomics? I get asked that sometimes. Well, it really depends on what you're doing. Targeted metabolite analysis, you may want to start with the triple quad. For simplicity and accurate mass analysis, a TOF is hard to beat. If you need accurate mass and some MS-MS capability, a QTOF is good. Highest capability, but with highest cost, perhaps an LTQ Orbitrap. Each instrument has its own advantages, as I already described. So available resources and your assay requirements will do this. The good news is the core lab, if they don't make that choice for you based on how you decide, is going to at least provide a lot of useful advice that will let you guys pick an analytical method that will be best suited to your analysis. So briefly, instrumentation in the MRC squared. We've got the instruments we need to run your samples. So we have 6 QTOF, 5 triple quad, 1 TOF. That's current as of my last count. I might be off by 1 there. I think that's added the new triple quad. Maybe it didn't. I forget. We've got 2 GCMS systems, 2 GC flame ionization detection systems, which are used for certain lipidomics type assays, and some automated sample handling equipment, additional sample prep resources, and support equipment. So you'll get to see some of these fun machines when you walk around the lab in a couple days if you haven't already. But suffice it to say, we have the equipment to do the assay that you need to do if it's metabolomics here. So in summary, metabolomics, like other omics sciences, is based on instrumentation. NMR and mass spec are complementary. Steffi will tell you more about this. And we've got the instruments we need, and I'm happy to answer any questions. And now we can close the black box if you guys are done. Yes. Okay, very good. I think, I mean, so yeah, it'll depend on the assay, right? if you're trying to replicate a study that's already done and it's a quantitative method and you're following a published protocol, then we can reproduce it, and that may be a triple quad. But yeah, I would definitely, in many cases, when we know that we can do just as good of a job on quantitation on an instrument that is compatible with untargeted profiling as we can on, say, a triple quad, then absolutely, I would often recommend that, why don't we do this? Why don't we set up to make sure that we can do your quantitation that you need, but let's also get all this other great data that you can go back to and mine and look at it in an untargeted way if you see fit, or run the untargeted assay, and then we'll just be sure that we provide the quantitative, targeted data that you need out of there. I think rather than jumping to a triple quad, highly targeted assay right from the beginning, especially if you're starting out an experiment, I would say, look at picking a method that can answer more questions, even ones you weren't thinking necessarily about asking, because you might discover that you want to follow up on something that you hadn't predicted. But that said, it's all a case-by-case basis, and it depends on what you want to do. But yeah, no, absolutely. I think a lot of the times the time-of-flight-based, untargeted compatible analysis is the way to go for a first approach, and will give you, just as good quantitative data as what you might get off a triple quad. Exceptions exist in all cases, but yeah, and we do recommend that frequently.
Similar to what's happening in the area of genomics, it looks like we're experiencing somewhat similar in metabolomics, where basically, whether it is an exome capture, except for the genome, whether it is an exome or a gene panel, there are no two gene panels that are alike. And the same thing is happening here for the core labs that are NIH funded, like Michigan's,
sure,
as well as the commercial vendors like Metabolon. Looks like the platforms on the surface, they may be comparable, but there are probably significant differences. And for a user, an end user, like a non-core person, how do we make the best choice?
Yeah, that's a great question. So yeah, and that's a developing issue in the field, is the idea of standardization and the ability to get the same sort of thing. So the easiest answer and a non-answer to your question really is, for a given experiment, the safest and easiest thing to do is to pick a platform that will have good coverage. And like you said, the platforms that are out there are generally comparable, right? You can take a time-of-flight-based instrument, or a separation method that will work with polar metabolites. And yes, there may be slight differences across sites, but the overall coverage is the same. And then pick one and then use that for your samples, right? It certainly helps as far as data consistency is concerned to batch all of your samples all at once, but and run them on a single platform so that you can do the relevant comparisons. Where it gets tricky and something that is really developing in the field is where you need to submit samples multiple times over the course of years, right? And you need to maybe compare data generated at multiple different sites and stuff like that. And so the way that is needs to be dealt with at this stage, unless you're dealing with it, so one option is to go to an extremely quantitative approach, right? Everything in terms of micromolar, nanomolar concentrations, using a calibrated approach, and then things should be consistent regardless of the platform if it's properly done from site to site. But in the case of untargeted profiling, that's much more difficult to do. So what we would recommend is that you talk to us, or you talk to whoever you do your metabolomics with, and establish a plan to allow the cross-platform comparisons to be able to be done. And a lot of the times, the best thing you can do is generate a pooled sample, or a sample that's going to be run consistently across multiple platforms, and that data will be acquired every time you run an analysis, and sort of serve as a frame of reference by which different methods, different platforms can be compared. You can look for the same identified metabolites, the same even unidentified metabolites based on their mass across platforms, and you can correlate them, and you can then do things if necessary to bring them up to the same level. And I've lately worked a lot more, not so much across multiple sites, but across samples that were acquired over a long period of time, at bringing them to the same level and normalizing. And there are, as long as the initial analysis is done appropriately, there are, are getting to be better and better techniques available for doing that. So my recommendation would be to, consult with the core wherever you do your metabolomics and make sure that you have a plan in place to standardize your runs across batches and across sites, and then get a good statistical plan in place to be able to do that normalization and look at things the same way. But you're right, the platforms are still somewhat different, and you may get a profile that's somewhat different across across sites, and there still is a way to go to bringing that all onto some sort of an absolute standard as a field, and whether we'll ever get there or not, I'm not certain. It's a good point, though. Other questions? I'm probably going over my time here. Very good. Thank you all.